So when Maggie invited me to give this talk, I was frightened because, it's high quality science and that I'm going to hear. And honestly, what we are talking about, our 5 cases, won't be that much. And unfortunately, and perhaps fortunately, my fears are confirmed. But nonetheless, that's what I'm going to talk. So how this thing works. How this thing works. Yes. So basically, it's coming from a cyto director. So if your only tool is a hammer, every problem looks like a nail. That's a thing that I always like. And you may ask, well, disclosures, all the experiments are performed by Bionano Genomics. I don't get any compensation. We didn't get anything. Although I got a very good gift box from Bionano today with that, but So why are we still doing karyotypes in clinical labs? Well, because it's a single-cell genomic assay. It's a whole genome analysis, and it is $30 to $50. So it detects copy number variants, it detects structural variants, it's low cost, and no reimbursement issues. But the biggest challenge for obviously karyotyping is the resolution. So this is where the chromosome microarrays stand for, right? It's whole genome analysis, it detects copy number variants. It's the compared to low cost, it's an acceptable cost. Particularly for cancer cases, there may be reimbursement issues, but it is accepted practice. I think optical mapping is the detection of structural variants is the main attraction for a clinical lab for potential use. And the question that we asked was, okay, is it feasible to use optical mapping in a clinical lab? So we sent 5 cases to Bionano, and they run the assay and do the analysis for us. And we actually run our results in the clinical lab. So we got 1 case of BALL, 1 case of multiple myeloma, 2 cases of AML, and 1 case of CLL. So for BALL, so why are we doing this? In a sense, in a clinical setting, because we use cytogenetic data to classify the risk, whether a patient is a good risk or a poor risk. If the patient has hyperdiploidy or ATV-RUNX1 translocation, that's a good risk. Is hypodiploidy and also BCR-ABL translocation or pH-like ella, it's basically a bad risk, and it is kind of important. So I would like to mention a particular pH-like BLA. I don't want you to memorize this slide. This is too complicated. But the bottom line is, there are so many translocations. You cannot do FISH for each of them. You can perhaps do qPCR experiments, but which are done in major diagnostic reference lab in US. And to my knowledge, I think there are only 2 at the moment. Also, there may be some NGS-based assays, like, for example, Archer is one of them. But again, you have to know what you are looking for. And whether you got pH like BLL or not is important, because if you got that translocation and it's not being discovered, your patient is going to die very soon. If it is discovered, the patient will get tyrosine kinase inhibitor, and hopefully will be treated. There's a very important clinical utility. So how do we work up a new diagnostic case of an adult case, for example? We use FISH for BCR, bone marrow smear, and KML and TP3 on culture. If flow is positive for CRLF2, we use with FISH. We process chromosomes ASAP. And based on the looking of the aberration, we order additional FISH testing during our workup. But our pediatrics is slightly different. Obviously, we do BCR, ABL, bone marrow smear, but also to identify the low risk with the ETV6 and RNX1. For all of our cases, we triage our cases. We don't run a case is not positive with bone marrow morphology or flow testing. And we tend to, particularly with the bone marrow smear cases, we try to turn out in 16 to 24 hours all the time, because the faster the results are, the faster the treatment. So the case that I'm going to talk about is a 18-year-old female, new BLL, plus 93%, which is very bad. Flow was CRLF positive, so we had run chromosome analysis and 46XX plus X on 7 cells. So and our fish was positive for CRLF2 rearrangement. So the issue as standing for our pilot is that, are we going to see the CRLF IGH translocation? So, okay, this is the X-ray X chromosome. And with our fish probes for CRLF, it's a break-apart probe. CRLF2 is in between. So we got the... what? So we got the green and red probes flanking the gene of CRLF2. And when they are together, they make a good yellow color. When there's a rearrangement, it makes a red and green and a red color. And this is the normal fusion products you see on the right-hand side. And the left-hand side, we got one green, one fusion. The fusion is here, and green, red, and red is here. So we certainly have the abnormality. So when we go to that, again, I would like to see those things labeled as X and Y, but it's my perhaps pet peeve as a human geneticist. But nonetheless, the assay was able to find the X14 translocation, which makes us very excited. And when it was followed, interestingly, CRLF2 wasn't reported. This does not encompass the CRLF2 gene. But one should not forget that, particularly the translocations, which involves IGH or IGHL regions, it doesn't necessarily cause fusion genes. So it's actually what it does is that the translocation causes the overexpression of the molecules. So, and this is the molecular support for that. And let me remind you again the probe itself. And this is the CLLF2. So breakpoints is somewhere in between. So it doesn't involve the gene, but it certainly causes the translocation, and the gene is overexpressed, which is also confirmed by flow cytometer studies, as I mentioned before. So for multiple myeloma, genetics of multiple myeloma is complicated, but it is mostly the initial IGH translocations, 614, 14, 16, 11, 14, and 1420 translocations. There are multiple translocations in multiple myeloma, and the disease has also clonal heterogeneity. So there's clonal evolution when you do that. Also, another challenge in terms of cytogenetics for multiple myeloma is that those cells do not grow. So most of the times you have normal. If you have an abnormality for multiple myeloma, it usually is not a good sign that it pertains either very active tumor or it pertains a high neoplasia load. And we use that information for prognostic classification, which also guides the therapeutic intervention. So the panel that we have is we run a We do FGFR3 and CCND IGH for translocation panels. And these CDK2, CK1SP is a bad prognostic factor. The PIF3 is like that. RB1 used to be, not anymore, fortunately, after the bortezomib-based treatments. But again, an assay like this has the potential to identify the translocations. So this is a 65-year-old follow-up multiple myeloma case, and unfortunately, the patient is deceased. And in our analysis, we got extensive structural and numerical changes. And FISH was positive for CDKN2C, S1B, and RB1. And NGS is also yields RAS pathway abnormalities, which by itself shows clonal evolution. And this is the karyotype. I mean, it's a horrible karyotype. But I mean, what you can see is hypertriploid karyotype, multiple structural and numerical abnormalities. And we have basically gain of CK1SP with those 5 reds that you see. There's a clone there. Also a clone with the deletion of CDKN2C and a gain of CK1SP. And also an RB deletion, both in a diploid setting and also aneuploid setting. Well, this thing wasn't that helpful. I mean, we know that it was complex, and it's still complex. But of course, we are looking at a perspective of a clinical lab. I mean, there is much more in this, under it. But what it shows, perhaps the underlying mechanism, recombinative events here. We definitely see the gain of chromosome 1 and deletion on chromosome 13 and loss of structural aberrations. And with the summary of the results, we got that 8-to-1 translocation and other, and all the aberrations was basically confirmed. So it is, we are quite happy, at least at high level of data. Okay, for AML, genetics of AML is very complicated, but the message that I would like to derive here is that structural variants are important prognostic determinants, and those prognostic determinants is not just for risk assessment. It's not just for studies. It also guides the therapy that the patient is going to get. So the favorable translocations is the RUNX1, RUNXT1 translocation, CBFB, IMH1, and adverse one is 6-9, any MLL translocation rearrangement except T9-11, which is an intermediate risk factor, so on and so forth. So what we do here is, in 24 hours, resolve the issue of CBFB and RUNX1, RUNXT1, chromosome analysis ASAP. We would also run arrays for AML, which will be done in our molecular laboratory. So our first case was basically a normal case. We got MPN1 duplication, which is associated with normal diploid phenotype anyways. And FISH is normal. Chromosomes are normal. And I think that was one of the questions was that, there isn't any overt rearrangement. There are a couple of structural variants, but that was one of the issues. Why? Because we used this case as a normal control for the study. So we don't want to deal with aberrations that's coming our way, which we don't know what to do, honestly. This case, the second case, the 26-year-old male, MPO positive plus, so leukemia RT-PCR panel is negative. So we got deletion of chromosome 1, 6, and there is additional AD7. So AD7 means that some chromosome of unknown material is replaced in 7p at that band and distally. We saw a ring chromosome and in addition 17. CMA gives us the deletion. Also, basically confirms the major cytogenetic findings. But interestingly, it did not show anything. It didn't say anything about the L16 that we were calling. But if you look at this, yeah, it very looks like deletion, but it may be perhaps this dark band is something else. This looks like a ring chromosome, but it may very well be something else. And there's also an AD17, which we don't know what they are. I think it confirms all the findings that we have at high level, but most interestingly is this so we got quite a complicated rearrangement, a 716 rearrangement on 617. So probably the deletion that we see is basically 617 translocation. For CLL, we do standard work, also arrays. CLL abnormalities are usually copy number abnormalities, but research-wise, there are some interesting translocations and also point mutations. So all these cases get NGS from the get-go, from the initial diagnosis, and most of the cases will also get arrays on top of cytogenetic analysis. So we got trisomy 12 here. That's the bottom line, and you don't need to be a cytogeneticist to see this trisomy 12 here. It's quite obvious. It's always here, and with the fish panel, you see 3 green dots here, and we got trisomy 12 here also. And then additional translocation with 517. I will not talk about it, but because we didn't confirm it by any other methodology. But it involves a gene which is quite interested in JAK-STAT pathway, which was reported in CLL, but buried in the supplementary data of various papers. So it was quite interesting. So our question, is it feasible to use optical mapping in a clinical lab? Yeah, we see all those abnormalities that we're supposed to see. So I think it is something worth investigating. Certainly we are encouraged, particularly with the CLF2 translocation. But There are also analytical issues that really needs to be, answered, and it was also brought out in various talk in this talk. For example, the ground truth. So, what is your very well characterized sample? I mean, that was the talk Tina from WashU. I mean, that's what the ground truth is. What kind of an orthogonal technology that the clinical lab use? I mean, we should really use whole genome sequencing or PacBio sequencing, or is just standard of care technology like, I don't know, chromosome microarrays or the standard NGS base are enough. Size of and nature of the aberration is also important. We are not reporting all the CMV cases in a microarray lab. We use judgment or cutoff according to the region. Limit of detection is an important issue, which we didn't formally address, but in a validation it needs to be addressed. Bioinformatics is still evolving field. There is still work needs to be done. Obviously, people are coming with their own algorithms, own solutions, which is great, but there needs to be probably more maturity in the field, which is evolving, don't get me wrong, and also how to tackle with novel findings. I think it's not perfect, but the things may be good enough to proceed, particularly for very well-known translocations or aberrations with known clinical utility. So any study needs to address those major issues while doing the validation. Of course, another issue is how to use this thing. Are we going to use it for initial diagnosis versus follow-up? Because I heard that it's going to be it will replace fish and chromosomes. Maybe, but if we identify the aberration, perhaps it's good to use fish to follow up that aberration. And also, the limit of detection of this assay is about 10%, which is a good assay, comparable to arrays, even better than arrays, some may argue. But most of our fish cutoffs is about 3%, so it needs to be considered. Also, is it a replacement test versus supplementary test? If it's a replacement test, that will make my administrators more happy because patients will spend less money. If it's a supplementary test, the business argument is hard to develop. Also, throughput and turnaround time are important considerations. Also, there are business-related issues. Coding. What kind of a code that we are going to use for this test if we are going to use it? Perhaps we can use array codes, perhaps we don't. And how does the reimbursement is going to work? Who knows? Conclusion, it's a very powerful technology that enables us to scan the whole genome for both numerical and changes and balanced translocations. But confirmation of additional abnormalities detected by optical mapping and further testing with large cohorts for validation is certainly warranted. And before I conclude, I would like to thank my colleagues, Dr. Guilin Tang and Dr. Zhenya Tang, my partners in the cytogenetic lab in MD Anderson. We are the 3 faculty overseeing the laboratory. And for Bionano, I would like to thank Andy Panka for data analysis, for Jeff Robertson to organize everything, and Maggie for the invitation. And this is also for research use only. It's not for use for diagnostic procedures. And it may be also an important issue, particularly if FDA comes into play for regulation of this testing. For the last 4 years, the government did not, but it may change its mind, and probably any vendor should have close working relationship with FDA if they want the testing to proceed in clinical use. Currently, it is doable in this regulatory environment. CLIA labs can do things whatever they want when they do extensive validations. But if the government says that, okay, you can only use diagnostic use, then we cannot. Well, thank you very much, and thank you.